Well, thank you. It's a pleasure to be here. And today, I just joined the Department of NanoEngineering in 2008 and as an assistant faculty member. And to give a little bit of an introduction about who I am and where I came from and why I became what I am. So this is a little bit of a blurb, and you can see just from the where I've been for my whole life is within California. I've never left California. And so I'm very happy to talk to a bunch of students who also are from California. And so just to give a little bit of who I am, I was born in San Francisco, California, and I went to high school there. I don't know if any of you go to the high school that I went to, which is San Francisco University High School. And then from San Francisco University High School, I graduated, and then I went to UC Berkeley for 4 years, and I actually majored in cell and developmental biology. And then I decided I really didn't like biology very much, and I really didn't want to be a doctor. And so for a long time, my parents had wanted me to become a medical doctor, and I realized I didn't really like biology that much at all. And so I decided to leave, and so instead of actually taking and going to medical school, for example, from UC Berkeley, what I decided to do was completely change career goals. And I decided that I actually wanted to major in some form of physics and some form of chemistry because that was what I was better at. Okay, so from there, I went to a really beautiful location called Santa Barbara, California, and I went to UCSD where I actually majored in both a combination of chemistry and material science and engineering. And I don't know how many of you know what material science is, but it is a field that is very, very broad, and it covers a lot of basic disciplines all the way from chemistry to biology to physics and so on. From there, I decided after I got my PhD, I actually went back to Cal for another year as a postdoctoral researcher. And then I decided that I actually wanted to try industry for a while. So I went to IBM Albany Research Center, which is here in San Jose, California. And there I actually worked there for 4 years. And I think most of you know what IBM is and what it does, at least in terms of the computer side. And I did a lot of basic science. This is actually a beautiful location in San Jose, California. You can't actually see the IBM site at all from downtown San Jose. And actually, the Albany Research Center is on top of a hill. And when you go there, you see a wide range of scientific things that are occurring, all the way from engineering and chemistry and physics, for which IBM is very, very well known for, as well as services. And so if you're ever interested in talking to me about what it is to work in a company like IBM, you can certainly do that. After that, after about 4 years there, though, I decided I was getting kind of tired. And I was getting tired of working in industry. And I wanted to actually work again with students. And I wanted to teach. And so I decided to leave IBM after 4 years. My pay was actually pretty good, though. And I went from IBM, and I came here to UC San Diego. My pay didn't change that much. And from here, I've been here since 2008, and I've been in the Department of NanoEngineering. And what you can see here is from my research interests, is that I have a very, very broad research background. And what I'd like to talk to you today a little bit is about nanoscience. And I think, again, most of you have heard about nanoscience, at least in some form. Whether it's from a gadget called the iNano, or it's just about just hearing it on the internet, or reading about it, or perhaps hearing your family talk about it. What I like to do is talk to you about why nanoscience exists, why it's cool, why we should be interested in, and what we might be able to gain from it in the future in terms of applications. And then I'd like to talk to you about, maybe a little bit if I have time, about what I do within the scope of nanoscience and how engineers might play a role in this field. Okay, so first things first, we have to sort of say what is nano. And I think most of you maybe at some point have heard of the term nano, and most of you have related it to a size. Okay, so some of you might actually have seen something like this, which is the scale of things. And I don't know if any of you have actually seen this chart before. But if you type in the scale of things or nanoscience on the web, you'll find at least 50 copies of this somewhere. All right, it's a very popular slide that a lot of people like to show, and the reason being is that it scales things in terms of things that you might understand. All right, in terms of both natural systems, okay, meaning living systems, okay, all the way from animals down to DNA, which is very, very small, and as well as things that are man-made, things that you can actually hold in your hands that somebody made for you, and to things that are actually on the atomic level, but also man-made. All right, so these are the things that people like to talk about when you type in the word nanoscience. This is what you usually pull up, is the scale of things. And the reason why everyone talks about that is because nanoscience is very, very small. We work with materials that are tiny. Okay, so if you think in terms of a dog, a small dog, and you say it's about 300 million nanometers, all right? DNA, on the other hand, if you've taken any sort of class in biology, you know that DNA is the genetic code of life, okay? It's what makes us who we are, okay? DNA, or it's called double-stranded DNA, is actually only 2 nanometers wide, all right? So as nanoscientists or nanoengineers or engineers who work in nanoscience, we work with materials that are literally 100 million times smaller than your pet. Okay? Now, pet you can obviously hold. So how do we actually work with materials that are really this small, and why should we at all? Okay? And so this is a thing that people like to show a lot because it actually shows all the ranges and sizes from both the nanoscale that we work with as engineers or as scientists working in the nanoscience area. And what we would like to do is figure out a way to actually build materials at all the length scales. Work with materials as small as this and make it turn into something as large as this. Okay? And the reason why we're interested in nanoscience is because there's a lot of applications that we are very, very interested in, especially as engineers. Okay, so nanotechnology, the marketplace, I don't think there's a lot of things that you can necessarily buy. The closest example you might think of, for example, is your laptop. Okay, that obviously holds your chip, which is made of nanoscale, has feature size on the nanoscale. Okay, but nanotechnology in the marketplace is a growing area, and there's a lot of people that are very interested in nanotechnology for a wide range of applications. We're not talking about just your computer chips or electronics or consumer goods. We're also talking about from the standpoint of solar energy. Okay, we should be very, very concerned about the energy right now, as well as health. Okay, and so in terms of applications, we have everything from consumer-based applications such as self-cleaning cars or self-cleaning paints, to electronics, to medicine. And so there's a lot of interest in nanoscience, but it's taken a long time to not only make a material that's small, it's actually taken a long time to build something useful out of a lot of these materials. And that's the reason why there's still a great need for us to work together as scientists to try to make something useful out of all this. If you think about it, there aren't a whole lot of products you can buy nowadays that are built from nanomaterials. Okay, and I'm going to show some pictures. Nanoscience, one of the nice things about working in nanoscience is you always have tons of pictures. And so if anybody wants, beautiful images, this is definitely the field to be in. All right, and so there's a lot of challenges within both the science and the engineering side as to how to take nanomaterials and make it useful for the marketplace, meaning for us to use and for us to appreciate. Okay, so this is a slide that actually Professor Vecchio, the chair of the NanoEngineering department, likes to show quite a bit. And the reason is that there's a new way of thinking about how to do engineering in the upcoming years. And so if you're interested in nanoscience, and if you're interested in a lot of materials that I'm going to talk about today, there's a growing field and a growing necessity for people to work together to actually make them useful. Okay, and so for example, for solar applications, we obviously would like cheap solar cells. We actually would like to find ways to take sunlight and turn that into energy as a renewable source, right? But how do we actually do that? How do we actually make a cheap solar cell? And if you're going to actually use nanomaterials to do that, how are we going to actually do that? We haven't really been able to do that as far as we need to get to yet. For medical applications, if you're interested in actually trying to design and help medical doctors figure out a way to get drugs to a very localized system within your body, how do you actually do that? How do you actually deliver drugs safely to your disease site, like a cancer cell, and not have it go anywhere else? Right? And so there's a lot of materials that are very useful, and I'm going to show you some pictures today. We're going to start all the way from the beginning. But we have a very long way to go to actually make any of the materials toward where we would like to be in the future. Okay, either for medicine, for health, for sensing, for fuel cells, for whatever your application might be. Ultimately, too, you also have to think about cost. I mean, that's the thing a lot of people are also thinking about nowadays, is how do you actually make a lot of these materials in large yields, still be environmentally friendly, you don't want to damage the environment, and then also be able to make it cheaply so that anybody can actually use these materials. Okay, so what does nano look like? And if you type in the word nanoscience, you'll find a ton of pictures. All right, I mean, these pictures are everywhere. And they come in all shapes and sizes. And the typical ones that many people like to see, or actually work with even, are nanoparticles, which basically are small particles. So these are particles that are oftentimes spherical. In this case, you can actually see one of these particles here. These are nano-sized particles that we can make in the lab. Okay? And I'll talk a little bit about why they're actually pretty cool. We also have materials that a lot of people also have heard about, maybe, and some of my own students in my research group work with this, which are called carbon nanotubes. And this is actually a schematic to show what a carbon nanotube really looks like. It's basically a bunch of rings tethered together, and each nanotube and the typical width of a nanotube is about 1 to 2 nanometers in size, and they're very, very long. Okay, but they're very small. What you typically see, and I'll show a couple slides about how you actually can see nanomaterials. This is a picture. This is called a transmission electron micrograph of a set of carbon nanotubes. And each of these little tubes, I think you can kind of see, is a single nanotube. Okay, and you can actually pattern nanotubes if you want to be clever, and this got a lot of attention because, of course, this is President Obama, and this is actually a series of multi-walled nanotubes standing vertically from a surface that they actually they created a micrograph of. These are triangular nanoparticles. These are silver, and these are little triangles, and I'll also show why those are very interesting. But all of these things you can actually make in the lab. We can make them, and we can actually use them. Okay, and they've gotten a lot of attention because of some of the unique attributes that they have. It's not just that they're small, and I'm going to go into that. It's also that they have very unique physical properties that make them very, very interesting and make us want to use them for certain applications. All right, but typically when you think about nano, oftentimes you pick up something like nanoparticles, carbon nanotubes, or nanowires. Okay, but all of these we can actually make using a variety of different methods. Okay, so why are nanomaterials cool other than the fact that they're small? All right, the reason why nanomaterials are actually really cool and actually have quite a lot of advantages over larger materials is because they have a set of properties that make them very, very unique. Okay, so if you think about nanomaterials, we're talking about sizes of materials they're bigger than a molecule. Okay, a molecule is obviously angstroms in size, so they're bigger than a molecule. But they're smaller than what you might think of as a bulk material. When we talk about bulk material, we're often talking about, let's say, material you can hold that's at least millimeters, for example. Okay? So nanomaterials are often in the size regime somewhere between a bulk powder material and bigger than a molecule. And what that happens with a lot of materials is when you make them in this particular size, you give them very unique properties that they typically would not have, either at the molecular level or as a bulk crystal. All right, and so a lot of the interest in nanoscience came about because not only did people make such materials, people also found they had these really cool physical properties that made them really, really, really interesting and useful to use for a wide variety of applications, whether you talk about it from energy or health. All right, and so just to give you an example of what we were talking about here, one of the most typical materials that nanoscientists like to work with are nanocrystals. And nanocrystals are typically made out of inorganic materials. And what we mean by inorganic materials is we're talking about metals. Okay, so if you look at the periodic table, and you should have seen this in some form in your chemistry classes, you can actually make nanocrystals from those materials. For example, such as gold nanocrystals, iron nanocrystals, platinum nanocrystals, and so on and so forth. And we've actually made a wide range of materials out of these inorganic materials. You can also do it out of mixed materials. So for example, if you look at the periodic table, you'd see cadmium and selenium. If you take the two together, you make a semiconductor material. Without even going into what a semiconductor is, you can actually make nanocrystals out of cadmium selenide. All right, and this again is just a TEM. Each of these little spheres is a single nanocrystal, and I have a couple slides to explain, how you actually image these things. But each of these little spheres is about 4 nanometers in size, and they're a single nanocrystal made out of these different types of materials. Okay, so what makes them so cool? All right, you see they're small. You can make a wide variety of shapes. What do you do with all this stuff? All right, one of the first things that people noticed, or that it gets gained a huge amount of popularity in the field, was that one very unique attribute that really proved that these nanomaterials have different properties than either the bulk materials or a molecule, is if you take a material such as this material here, cadmium selenide, again, it's a mixture of cadmium and selenium. If you just buy it as a powder form, cadmium selenide, there's nothing interesting about it in terms of light. Okay, everyone likes colors, right? So if you take a look at bulk cadmium, and so you can see that here, cadmium selenide, right? This material by itself, if you shine light on it, nothing happens. You don't see anything, all right? So this is what I mean by bulk. It's just a large powder material, okay? However, if you take that bulk material and you make it really, really small, all of a sudden you see a wide range of colors emitting from this same material if you shine light on it. So why is that? Why do you see all those colors of light? All right? And the thing is that you can actually tune its color, okay, simply by taking the same bulk material and actually making it really small. And you can make any type of color you want in any sort of, in a very tunable fashion, okay? And without really going into all the science behind it, the main reason why this actually occurs is because of the fact that you actually change the physical properties of this bulk material in such a way that it now emits visible light, okay? And if you're interested in any of that kind of physics, which you'll learn someday if you decide to work in, especially if you decide to work in nanoscience, is that you can actually tune the physical properties of these materials that instead of a bulk material that shows no light whatsoever when you shine light on it, okay, there's a reason for that. This material, on the other hand, you change its energy states such that you can actually emit visible light. So you shine a light on it and you'll see all the different colors, all right? And the way you actually change the colors, you just change the size of the particle. Okay? Now the reason why that's so exciting, all right, is not just the fact that it's really cool physics and it's really cool science, all right? And it's not just the fact that we can make really small things. We like that too, all right? But there's a real application for this, okay? And the application has to do, if anybody here is interested in biology, the application has to do with the fact that these are really, really small particles that all emit a certain color, and you can tune that color. All right, and that color comes from the fact that the color that these particles show, okay, so this is a solution of different vials that you see here, okay, they look colorless, right? Each one of these contains lots of these, all right, so each one of this contains different solutions of each of these little tiny particles that are around 4 to 6 nanometers in size. Okay, if you shine UV light on all of these little vials, you actually see all these different colors. Okay? And all these different colors is very tunable. You just change the size of your particle. Okay? So why is that interesting from a biology standpoint, from an application standpoint? The interest of these particles is that these particles now are so small that they're actually the same size as a protein. So what does that mean? That means that if you're interested in imaging a cell, okay, I don't know if anybody has ever taken a biology class and has actually imaged cells or have seen cross-sections of cells in biology, you can actually use these materials to label living things, like cells, okay? And so the reason why this gained such a huge amount of popularity was because of the fact that these particles can be easily tuned in size, they're small enough to enter any cell you want, and you can simply tune the light that gets emitted by tuning the overall size, and the difference in these particles are not that large. All right, so from trying to understand how life or how living systems work, what types of proteins exist in a cell, how we can actually use these things to understand where drugs go, for example, in a living system like a mouse, these diagnostics became really, really cool to use. All right, and it's very, very neat in the fact that all you have to do is change the diameter of the particle you actually make. All right, so this is the one application where people got a lot of interest in nanoscience. They said, "Wow, this is really cool. You know, I just make a different size particle. I can emit now a different color, and I can use that, and I can, you know, put stuff on that particle, and it can label a cell, and I can tell what's where in that cell." Okay, whereas typically they could not do that very easily before. The other thing about particles is that they not only emit a photon or emit light, they actually also scatter light, meaning that if you hit a particle and it has a distinct shape to it, it'll scatter the light. So that light will scatter if the particle has a different shape, it'll scatter light differently. So what that means is that if you tune the shape and type of material of your particle, you can actually see different colors. Okay? So the reason why nanoscience is really, really interesting is, again, it's not just that they're small. I mean, it's nice that they're small because then we can actually use it for certain things. But on the top of that, they have these really cool properties. Okay? And what you're seeing here are the optical properties. And these properties we can easily tune simply by how we synthesize it. Okay, so we make all these materials in the lab. All right, a lot of these materials are not hard to make, and you can actually. I think we're trying to design certain courses that exist here for undergraduates at UCSD, where they might learn someday, they do actually laboratory research to learn how to actually make such particles. Some are harder than others, but some particles, such as the ones that are being shown here, are actually quite easy to make. You can make them in water. Okay, and you just take some silver salt and water and you shine some light, and then you find that these particles actually emerge. Okay, so they're really not that hard to make, and yet they got all these cool properties that we can also use. Okay, it's one thing to also talk about particles. You can actually, so far I've only really talked about nanoparticles. Obviously, I could also talk about carbon nanotubes and why they're cool. But it's not just the material, okay? You can actually also tune a surface in such a way that if you make it nanostructured, meaning that if you put some structure into that surface, okay, so if you think about your tabletop, right, you can imagine it's pretty flat. Okay, let's say now you put these tiny, tiny little grooves in it. Okay, really small grooves. What scientists have found is that if you do that, you can change its surface properties entirely. All right? So nanosurfaces has also gained a lot of attention. And again, the point of all this is that if you put some nano component into your surface, you change its properties very different than it would look like normally. Okay, so one thing that people found is that if you actually take a surface and you put like little tiny nano grooves in that surface, you can actually make a water droplet sit perfectly, just round up, just bead up perfectly, and it'll just roll off. All right, so the water won't actually wet the surface at all. It just beads up into a little ball and just rolls right off. All right. One reason why that became very, very popular is this, they termed this as superhydrophobicity. The reason why they became very, very popular is people thought of ways to actually have self-cleaning paint. So you spray some water on it, water just rolls right down, collects all the dirt along with it, and there you have your super clean surface. It's a very cheap and economical way to clean things. People have also been thinking about ways to do this with their own clothing. All right, so superhydrophobicity, and all you do in this case is you make little tiny grooves in your surface. That's it. All right, and so another one is what some of you might have seen. I'm sure. Actually, I know all of you have seen these at some point, are geckos. And these geckos have these little pads. They have these little feet. These things can climb walls, right? So people were really interested in how do these things climb walls? What makes them be able to cling onto a wall, retract, go up, and keep going up? Okay, and if you look at their little feet, they actually have these little nano grooves. Okay, and that gives them really cool mechanical properties that people became very interested in. And so there's actually a lot of scientists that are actually trying to mimic this gecko because of that property. All right, so nanosurfaces is another way to think about it. You don't have to talk about just materials like what you make in the laboratory, and that's cool, they shine light and all this stuff. You can also think about it in terms of actually changing the surface of your whatever material you're looking at. All right, so how do you see nano? Okay, so a lot of things that I think that, and this is again sometimes a challenge when how do you educate students, is because oftentimes students really want to see what they're working with, and you can't actually see these things. All right, so the only way to really see nano, and I think at some universities which they're starting to learn or trying to have laboratories also built within so that students can actually learn how to run these same machines to actually see such nanomaterials. The only way to really see a nanomaterial is to use an electron beam. Okay, nanomaterials are obviously small. Okay, you can't look at it and use a light microscope to see it because light is larger, it's a few hundred nanometers. Okay, so what you need to do is you need to use an electron beam. All right, and if you ever get a chance to actually see one, it's a lot of fun to use. Okay, and typically when people first start, they look at flies or they look at bugs, and you can actually see everything. And the main way you actually do this type of electron microscopy is you either do it by looking through a very thin section, all right, or by actually doing something called scanning electron microscopy, which really looks at the surface, okay? And so again, if anybody's really interested, these are the two main ways, and there's other ways also, and I think as time goes on, as you take more and more classes, you might learn on some of these other methods. There are machines out there that allow you to actually image nanomaterials, to look at nanosurfaces. So we're not just, all these pictures that you're seeing are actually things we've actually taken pictures of, but you can't use just a bright optical microscope to see these things. But if you use electron microscopy, you can get incredible resolution. And so you can see things all the way from a the eye of a fly. All right, and so this is a zoom in of the fly's eye. You can see things such as pollen, but you can see everything. All right, and depending on how on the resolution of your microscope, you can get down to at least a few nanometers sometimes. Again, depending on what you're doing. All right, so basically I've talked to you a little bit. This has been a very brief overview of nanoscience and why. There's one example about why you might be interested in nanoscience. But as an engineer, I think what I want to show a little bit is about some of the work that I do as well, which was I have a lot of interest in basically how do you take all these nanomaterials and make an application out of them. Okay? So what that means as a nanoengineer, I am in the Department of NanoEngineering, is if I make all these small materials, and they don't have to be these inorganic man-made materials, they can be natural, biological. Okay, and I make all these materials, I have all these materials in my hand. How do I make something that's useful? How do I make a solar cell? How do I make a transistor? How do I make a sensor? How do I make something you can use for medical imaging or medical or delivery? All right, so this actually takes a lot of engineering. All right, and the main reason, okay, is in this part has to do with the size. All right, these things are very, very small. Okay, I can't pick it up with my tweezer and put it where I want to, right? So I need to figure out ways to take these materials that I can't even see in the optical microscope, somehow and put it together into something I can hold and deliver, okay, and have it useful. And that's actually really challenging, right? I mean, can you imagine how to do that? It takes. So we need to work together as scientists to try to figure out how to actually take all these small little things that have all these cool properties and somehow put them together into something that you can maybe use at home. All right, so one way that there's been some solutions on how to actually build nano features, and actually this nanoscience has been a term that's been used and gained a lot of popularity, but actually people have been working with nanomaterials for a while. So one way that people have gone around this issue about how to actually take small things and put them together into functional devices, one way to get around this problem is to actually use a technique that's called photolithography. Okay, and photolithography is a conventional way that they use to build your semiconductor chips, your microchips, your computer chips. It's been around for many, many years, and it's the tool that's being used to make all your little gadgets now. Okay, so photolithography is one technique that exists that allows you to actually build devices that have very, very small features. All right, and this is something that's been around for many, 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 many years, and that's the reason why we have all the gadgets that we have today. All right. So if this wasn't around, we would have a very hard time doing what we're doing. I would have a very hard time showing this presentation. All right, so photolithography is it's a known technique. It's been around, and I just want to mention because it's not like we're nanoscience has been around for certain applications for a very, very long time. And in this particular case, people have used photolithography to create your computer chips, all right, or to create devices or silicon-based devices. And the typical way you think about photolithography is you have some sort of polymer, you shine light on it, you have a mask on it, and that allows you to actually make your features that you can then use to make whatever feature you want out of silicon. All right, so this is a very, very well-known technique. If anybody has any parent who's working in this field, your parents might have talked about this at some point, where you actually can use photolithography to create very small features, and for electronic circuits, this has, again, been around for some time. However, there's been a problem with lithography, and that's the reason why I'm here, and that's the reason why a lot of us are still working in this field. If photolithography could answer all of the, give us all the solutions that we needed for every technology, then we really wouldn't have a job. But the thing is that photolithography has had some challenges, all right? And one of the major issues is that if you take a look at how photolithography is done, there's a size limitation, meaning that the features that you can create by photolithography, they're trying to get below this, but it's around 45 nanometers, okay? We work with materials that often are on the order from 2 to 5 nanometers. So I can't exactly use photolithography to do what I want for a lot of applications. The other problem is that photolithography works very, very well for silicon. If I'm working with materials that don't have anything to do with silicon, I can't really use this technology that easily. All right? And third, it's expensive. As you try to actually use photolithography or any sort of lithography, below this size, the cost is going to go up. And so it's not the most economical way to do things. And so, because of all of these issues, people like myself we're beginning to look at other ways to basically take starting nanomaterials, like the ones I showed, and somehow put them together without necessarily having to use an expensive tool like photolithography. All right, and so one way that people are beginning to look at things is they're trying to understand, how do I take small objects that are about 5 nanometers, mix them with something, and get them to organize into a device? Okay, there's actually quite a lot of applications. People know what they want. It's just very hard to have us figure out a way to actually take these materials and somehow organize them into these device architectures that we really, really would like. All right? So one thing that people have done is they've actually taken a look at how nature does things. Okay? Because nature is the best nanoengineer. Is absolutely, nature can do things that we can't do at all. All right? And so one of the reasons why we're actually looking at how nature organizes materials is because we're hoping to draw some inspiration. All right? Again, we're working with materials that are around 2 nanometers. We take a look at that 2 nanometer material, and we want to organize it to something like something I can hold and take home and use. But I don't know how to do that. On the other hand, nature is really good at this. All right? So if you walk down the beach and you look at your seashell, all right, and you take a really close look at that seashell, what you'll find is this is an abalone shell. I don't know how many people have actually seen or held an abalone shell. You can certainly find them in plenty of places around here. If you take a look at an abalone shell and you basically take a really small, just look really closely at the shell of this abalone, what you'll find is this abalone shell is composed of many, many, many crystals. All right? In this case, the abalone actually uses calcium carbonate, chalk. All right? It's a very easy salt crystal to make. All right? But what the abalone has done, all right, is it's actually taken all these little single crystals of calcium carbonate, all right, that are small, all right, and organized it into something huge. All right? So if you take a look at this entire abalone from here to here, what you'll find are all these little crystals of calcium carbonate organized in what is known as a brick and mortar fashion, because it looks like bricks and mortar. Okay, the mortar in this case is some sort of protein glue. All right? But the abalone has been able to self-assemble, and self-assembly is a term that we use a lot in this field, which it means is take a lot of things, put them together, and have them organized into something that has some degree of order. All right, so in this case, the abalone has actually taken some single calcium carbonate crystals and somehow organized it into these sheets of calcium carbonate, all right, to make a really, really tough shell. In between each calcium carbonate layer are some proteins. All right? The abalone was actually really, really smart. It was trying to make a shell that was really, really, really mechanically robust. All right? So how did it do that? Out of chalk. All right? If you take chalk, you can crumble it, right? So how do you actually take this? How do you make a calcium carbonate shell really, really strong? You take a layer of calcium carbonate, you put some protein or organic glue, you put another layer of calcium carbonate, another layer of glue, and you hold the whole thing together, and all of a sudden you can't crack it. All right? It's got some organic glue holding it all there. All right? So this is really, really smart on the abalone's part. All right? But this is the type of stuff that we look at because we're saying, hey, nature's pretty good at this. Nature's really good at understanding how do I take small crystals and put it into something huge for its function, which is to stay alive. All right? You yourself have this same component, your skeletal structure. It also is composed of a lot of little crystals that are held together by some protein. All right, so and obviously we function, we live, we walk. So so in nature is usually function driven. We create because we need to live. All right, and so because we have the skeletal structure, we can actually walk. We can actually stand up. And if you take a look at its organization, though, again in the SEM, in the scanning electron microscope, right, it's the only way to see at this type of resolution. You can see all these little crystals that are held together by, in our case, also protein. all right? And this is what makes up our framework, okay? So as nanoengineers, we say, all right, I have no idea what I'm trying to do, but I know I need to take some material and organize it somehow so that it becomes useful, all right? So we take a look at biology often for inspiration. Now, in my case, not only do I take a look at biology for inspiration, I actually use biology to help me to do what I want to do. So their concept then is this. Instead of actually using lithography to build a material, because I can't, okay, what about if I am smart and I think about like how nature does things? Is there a way that I can take my little materials, like my carbon nanotubes that are only 2 nanometers wide, or some particles that are 2 to 4 nanometers large, and somehow ask them to arrange themselves in such a way that I have my device? All right. So what we want to do, and this is basically an approach that we've taken in my laboratory. This type of research happens in many, many labs, where we're actually taking all these materials and actually getting them to somehow self-assemble. Again, self-assembly is a term we use to describe how things get self-organized. All right. And the way it works is you need to have something to drive that organization. All right. And so there's a wide variety of ways to think about how to do this. Okay, you can use a wide variety of materials. In my case, and in my research, I like to use biological materials to help me organize nanomaterials. And we work with all kinds of nanomaterials in my lab. All right, just to show one example about how hard this actually is and why it's taken so long, a really long time, is if you single-walled carbon nanotubes. Okay, so if you really want to see what a nanotube looks like, this little thing here is a nanotube. All right, it doesn't look very interesting to look at, but it has really neat properties. So this little nanotube here was discovered in 1991. And we haven't really made much from these nanotubes yet. All right, and the main reason is that it's so hard to actually both make these nanotubes the way we want to, as well as organize them the way we want to. And just to show an example about how hard this actually is, I think this is about 10 years ago or so now, one way that people were trying to organize these carbon nanotubes on a surface was to actually push one nanotube at a time into place. Like take a little tip, push it, straighten it out, and turn it, and then get it to where you want it to go. Now that's obviously okay for one study. That's very, very impractical in the long run, okay? But these things have been around for a long time. So why is it? It's been so hard, all right? And it's taken a long time to get where we need to go. But we're getting there. We've made a lot of progress in the years since. Okay, so the research that I focus on is on the use of biological systems to help self-organize materials, okay? And this is something I doubt you've ever seen in a textbook. I really doubt you've ever seen this in a textbook. And this is definitely a new field. Okay, we're very interested in using biological materials to help us self-assemble nanomaterials into particular devices. Okay, and sometimes it's easy to think about devices in terms of, that can be any device you're thinking about. If you're talking in terms of solar cells, or you're talking in terms of sensors, or transistors, whatever you want to think about in terms of your device. But basically, what I do in my own research is I look at how I could use biological systems to help organize these materials, okay? And so one way to think about that is that if you're talking about a computer chip, all right, and you say, I want to organize my materials, whatever my nanomaterial might be, into something like a computer chip, okay, I need to figure out a way to do that. Right? I need to take these really small things, somehow put them on a surface, and have them all perfectly arranged at the end of the day. All right? And so what I do in my own research is we actually focus on the use of biological materials, which is just drawn as a cartoon here, to help us get there. Okay? So what that means then is I take a big thing that's, let's say, 25 millimeters. It's large, a silicon wafer, for example. And then I use something in between to help organize something even smaller, okay? And so what my research group does, or a portion of what my research group does, is focus on the use of such biological materials to organize things even smaller. And I'm going to show you today some examples of what that might be. Okay, and the material we really don't care right now, Okay, this can be any types of materials. These are materials, pictures of which I've shown you. People have a lot of interest in this. What we're focusing is, how do I take these materials and organize them over an entire chip? All right, so what we use are biological materials. Okay, there's a lot of reasons we are interested in using biology to help us get to where we want to. This is, again, something new. And I think it's obviously different than what you've heard about before, because when you take biology in school, you learn about biology in terms of life. You learn about it in terms of genetics, cell biology, animal physiology. I don't think you ever hear about the use of biology and how it might be useful for nanoscience. Okay, but there's really cool things about biology that make it really, really useful. Okay, so this is a little bit of a stretch of an imagination, but there's a lot of people working in this field about where they're actually taking materials such as DNA, which you've learned is a genetic code, okay, that makes you who you are. All right, and DNA is double-stranded in its natural form. All right, and you have these bases that recognize each other. And again, this is just double-stranded DNA. Okay, this is the foundation of genetics. So some people are actually very interested in using DNA to help organize nanomaterials. Okay, other people are also interested in using other biological materials such as proteins. All right, proteins are often parts of a cell. Okay, some people are also interested in using cells. And what might sound even crazier than that is that people are actually also using viruses. Okay, viruses can be dangerous, but there are many forms of viruses that are not dangerous. Okay, and people are actually using viruses that can come in a wide variety of sizes and different shapes and are composed of many, many proteins. And they're actually using these viruses to build materials. And recently, there's a group at MIT that actually used these viruses to actually build batteries. All right? So there is a wide range of this type of research going on, which does sound kind of nutty, but the reason is that there's a lot of unique things about biology that we don't currently have, and so we're studying that to see if we can actually help nanoscience. Okay, in my own research group, we focus a lot on DNA and the use of DNA, and the reason is that DNA has some very interesting inherent properties that make it very, very useful to organize other materials. All right? So the typical form of DNA is this double-stranded DNA, which is just a helix. Okay, it's just a linear strand of DNA. You can think of it as a linear polymer. Okay? Now, DNA by itself is very interesting. All right, and one main thing is that it can hold a lot of information in its genetic sequence. The other reason why DNA became very, very popular for nanoscience, especially in the last decade, is that because you can engineer DNA to fold into any shape you want it to be. All right, that's what makes it really, really neat. So the typical form of DNA is a helix, meaning it's like a ribbon. Okay, it can be long, it can be short. Nowadays, you can actually engineer DNA to fold into all kinds of shapes, all right? Whether it's a cube, it can be both two-dimensional and three-dimensional. Whether it's a cube or some sort of octahedron or a cross or whatever. And it's not that hard to do, okay? And I'm going to show you some pictures to prove this to you. So one of the earliest examples, this started, I don't know, a while ago now, more than a decade ago. Where the first thing that people did is they said, could I engineer DNA to fold into something really unique, like a two-dimensional lattice? Okay, so they actually did that. They took 4 strands of DNA. The DNA is just color-coded here to show you what it looks like. Okay, each of these little lines here is a base. All right, and in DNA code, the adenine base always binds to thymine, guanine always binds to cytosine. Okay, so if you design your DNA correctly, these things will match up such that the red part here will hybridize to the blue part here will hybridize to the yellow part here, and you can actually get this thing to just by mixing these 4 different strands of DNA. You heat it, you cool it, and you get, this is again a picture to prove it to you, you get your two-dimensional DNA lattice. Each one of this is a DNA strand, and they're crisscrossing. Okay? Now, more recently, it got even crazier, and this is the reason why it became really, really popular. Okay? And this is because Dr. Paul Rothman at Caltech in 2006 found a way to get DNA to fold into any shape you want it to do, whatever you want. All right? And it's basically like having a CAD program. You use this computer program, you say, I want a smiley face, it'll spit out what you need to do to turn your DNA into a smiley face, and that's the reason why it got a lot of popularity. And the other main reason it got popular was because it's so easy to do. You can do this, actually you could do this at home if you had all the DNA strands. You could just heat it, cool it down. If you could image it, okay, you would find that you can make these things very easily. So what Paul Rothman was able to do, and again, he's a senior research associate at Caltech, is he was able to take this long piece of DNA from a virus and get it to fold up into whatever shape you wanted, and that's why he called it origami. All right? And so these are squares of DNA, triangles, a happy face, another triangle. You can actually write DNA in DNA. All right? So if you draw the map, the world, you can draw the map all out of DNA on DNA. And so that's what he's done here, okay? And you can write all kinds of stuff on top of that. This is really versatile. People have made all kinds of stuff since this. They've made the map of China. They've gotten a dolphin whose tail actually moves, okay? They've done three-dimensional boxes. So you can do whatever you want, and there's been a whole lot of papers since 2006 in this field alone. Okay, so this is what we're using as our current, and I'm saying there's a lot of advantages to using DNA. And the main reason being that you can actually use DNA very, very easily to position other materials. And it's really not that hard to do. And that's why it's very, very promising. And so although carbon nanotubes got invented in 1991, there is a lot of hope that we might someday be able to organize the carbon nanotubes or nanoparticles or nanoart or whatever you want into specific devices that you're interested in. And again, without going into all the details, I want to end here. We're working very hard at trying to figure out how to attach nanomaterials to DNA, how to actually, and you can use all kinds of ways to do this. And it's easy to buy, it's easy to do. And I'm making a story that sounds very simple, and it's not, but we're definitely on our way. All right, my own research group has done a lot of work to figure out how to organize these little DNA structures where we want them to go. So this is the dream that we had before, is you have a whole silicon wafer that's large. You somehow direct where these DNA origami go, and then you use the DNA origami to direct things that are smaller. We've done a lot of work to figure out how to get each of these are the DNA origami triangles that I showed you, how to pattern them, okay, on a silicon surface. That's step number one. And then we've also done a lot of work on trying to figure out how to get nanomaterials on this DNA origami. Again, this is just another picture to show you what you're looking at here are these little DNA origami patterned on your silicon surface. Okay, you need to do that if you're going to build devices. And we've done a lot of work to figure out how to actually get DNA to adsorb to a surface and how to get it to organize. And we've also figured out ways to attach things to these DNA origami, whether they're these 5 nanometer proteins or these 5 nanometer gold nanoparticles, which are these bright little spots that you see here. And so we've actually done a lot of work in my own research group to figure out how to interface DNA with materials that obviously are not DNA-based and are not even biology-based. And we've been able to organize gold nanoparticles using these DNA origami over a fairly large area. And this has taken us a lot of work. And if I remember, as I showed already, it's taken a lot of years to get us to go all the way from the synthesis of the first nanomaterial or sets of nanomaterials to actually trying to organize them. And one of the major reasons realistically was because we were looking for some discovery like Paul Rothman's, which he did in 2006. And since then, there's been a huge amount of work in this field, and we're also working on it very heavily in my own research group. And we're talking about all kinds of different ways to actually organize materials, and we're working really hard. And if anybody always wanted to come visit, come talk to me, you're certainly more than welcome. And my graduate students will be happy to show you. We can't show you the nanomaterial, we can show you what we're trying to do in the lab. Okay, so in my own research group, we are working a lot in nanoassembly. I have students, researchers in my group, ranging from material science and engineers to chemistry students. They came up with no biology experience, and they picked it up really, really fast. So it is possible to come with no biology, no chemistry, and you can pick up a lot of this type of research. I only talked up a little, about one fifth of what my entire group does, but we are very interested in this area of biomolecular assembly, and I'd be happy to talk to anybody more about some of the research opportunities as well, and graduate school, or sorry, undergraduate school, and what you might want to major in, and I'd be happy to answer any questions.
Thank you very much. Very fascinating talk. So we have time for a couple of questions. There.
Hi, my name is Christine. I just had a question on the part you were talking about nanoparticles, where you can tune it to tag proteins in cells. Is that material the same as fluorescent proteins?
No. So that material, so the green fluorescent protein, which was discovered in part by Professor Chen at UCSD, is protein-based. This is a counterpart to it. You could also use green fluorescent proteins or all the other fluorescent proteins for imaging purposes as well. There's a little bit of some advantages with the inorganic component, but this is very similar to that, where we make these particles that glow, and then we attach stuff so that directs it into a cell. His work is also very similar, and there's actually advantages to his work as well. But this is kind of the inorganic counterpart to his work.
Why is it so easy to tune the nanoparticles?
Why is it easy?
Yeah.
It's just a synthesis thing. So it's if you change the amount of, I think the amount of capping ligands. So basically the way it's made is you have some cadmium, or at least in cat selenide. You have cadmium, you have some selenium, and you have high temperature, and you have some organic ligands. And if you tune all that, you can tune the sizes really, really easily, and very, very accurately, actually. There is some work going on in a lot of different fields. Not all particles are that easy to tune. Some take a lot more work than others.
All right, I actually have 3 questions. Okay, one, will the cadmium selenide, is that going to be damaging to the proteins in the cell like erythidium bromide does to DNA?
Yeah, no, that is a very, very good question. Now, cadmium selenide is actually, that's been one of the reasons why it's not going to be really used for in vivo which means we're not going to ingest it anytime soon. But cadmium selenide is toxic, right? So people are working really hard to try to figure out ways to coat stuff on it to make it non-toxic. However, because whenever a person hears the word cadmium, they don't want it. I wouldn't want it, right? So the typical applications for this has been for imaging cells outside of the living system. But you're correct. It is very, very toxic.
And my second question is, does the protein, it was the protein on the that binded the calcium, the albigone shell. Do you have the name for that? Because that's very interesting.
I think they worked really hard to extract proteins from the abalone shell. I think they have one, and I think it's called lustrin. I don't know why they called it that. Oh, the luster, the pearl luster. So it's called lustrin A. And if you're interested, we have the whole sequence of the protein as well. But it's been really hard to get all the proteins from the abalone shell. The main reason being that the only reason to dissolve the abalone shell is to put it like a nitric acid. And that damages all the proteins. So it's been challenging.
Okay, and my last question is, before the electron microscope, we weren't able to go to the nanoscale. We were limited to kind of the large-scale micrometers and such. Is it possible with new technology to go beyond the nanoscale to get even smaller?
Well, we have atomic force microscopy, which claim is you can get down to, well, no, we can see at the atomic level. There are tools that allow you to see at the atomic level. They're not routine tools, though. That's one of the major issues with that. They're pretty special tools in order to do that.
Thank you.
Okay, maybe one last question. Okay, over here.
I was just wondering if when you're changing the shape of the nanoparticles, does that drastically change the function? For example, like the smiley face nanoparticle, as opposed to like the triangle one.
All right, so for the DNA origami in that
Yes.
case, I think he just wanted to make shapes. But yeah, it depends on what shape. So if you're trying to pattern for, let's say, devices, you want to make devices, the shape we would choose would be squares, just because it's easy for us to work with. The shape there's no function with his DNA. It's just to be able to create and fold DNA the way we wanted to fold it.
Okay, thank you.
Okay, thank you very much. Yeah. So here's giving back.